Dear children, today we are discussing with another basidiomyces fungi, that is Puccinia. It is coming under the kingdom fungi, division Eumycota, subdivision Basidiomycotina, class Teleomycetes, order Eurydiales, family Pucciniaceae. Puccinia graminis causes black stem rust. It is a heterogeneous fungi. Life cycle completed on two different hosts. The first phase is a dikaryotic phase. It is completed on wheat plant, so wheat is known as its primary host. The second phase is a haplophase and it is completed on barberry plant. So barberry plant is known as the alternate host of Puccinia. So Puccinia, heterogeneous fungi, life cycle completed on two different hosts. First phase is a dikaryotic phase. It is completed on wheat plant. Haplophase, second phase, it is completed on barberry plant. So barberry plant is known as its alternate host. Full life cycle is completed when both the host plants are present. Dikaryotic mycelium is unable to grow on barberry plant and haplophase is unable to grow on wheat. And coming to its life cycle, the first phase is the dikaryotic phase. This phase consists of a dikaryotic mycelium, two spore stages, that is uridinal stage and teleal stage. Dikaryotic mycelium reproduces by sporulation. Two kinds of spores are produced. First spore stage is the iridospores and the second one is the deltospores. And coming to uridinal stage, after few days of infection, dikaryotic mycelium begins to form iridospore below epidermis of wheat. Each iridosporus has a group of iridospores. Formation of large number of iridospores causes the cracking of epidermis of the host plant to expose the spores. After falling suitable host plant, it germinates by forming the germ tube, that is wheat plant itself. Later forms apricorium on reaching the wheat leaf stomata and causes secondary infection. Seen as reddish brown pustules on leaves and stem, hence called as a red stage. And this is the iridospores of Puccinia. Here it is a single cell spore stage and it has double wall layers. Outer wall layer has exane ornamentation. Outer layer is known as the exane, inner one intane, and the exane has ornamentation. Spores are binucleate, oval or round, produced on long stalk and have 4 germ pores. Here also we can see the scanning electron micrograph of these iridospores. Here we can see the ornamentation, exane ornamentation. Here also we can see the iridospores. And the second phase is the teleal stage. Later in growing season, same hyphal mass produces telea or teltosauri. This stage is known as the black stage. So, red stage is the iridosaurus stage and the black stage is the teleal stage. Initially, deltospores produced among iridospores. Later, sauri contain only deltospore. Late in the growing season, we can see the deltospores in the sauri. Sauri now called as deltosauri and the stage is known as the teleal stage. Spores exposed when host epidermis breaks open. 2 nuclei in each cell, that is, deltospores are bicelled and in each cell we can see a binucleate cell. So, each cell is binucleate. Later in the growing season, the 2 nuclei, the dikaryotic nuclei, fuses. So, the dikaryotic mycelium produces the teltospores. The teltospores has 2 cells and each cells at initial stage are binucleate or dikaryotic and later they fuse together to form a diploid nuclei. Here also we can see the cluster of teltospores which are 2-celled, dark brown or black in color. So this spore stage is known as the black stage or black spore stage. Here also we can see the teltospore picell stage. Here also we can see the teltospores. Outer wall layers are mostly thick. Here we can see the sori mixed with the iridospores and the teltospores. And next stage is the basidial stage. With the return of favorable condition, the teltospores germinate. Each cell of teltospore produces a promycelium that comes out of the germ pore. Diploid nucleus passes into the promycelium, divide meiotically, forming 4 haploid nuclei. The 4 cell structures separate by one another by cross walls called as the basidium. So, this is the first stage of the teltospore. Here we can see the bicelled dikaryotic nuclei in each cell. One is plus and one is minus. Later in the growing season, the 2 nuclei fuse together and form a diploid nuclei. And at the stages of germination, each cell produces a promycelium. At later stage, it becomes septate, and at the same stage, the diploid nuclei undergo meiosis and it produces 4 nuclei. The nuclei passes into the promycelium. Later, it undergo septation, and each nuclei with 1 haploid. Each cell has 1 haploid nuclei. And this nuclei moves into sterigmata or short stalk, which swells up and produces the basidiospores. Basidiospores are haploid and they are borne on short stalks or sterigmata. Here also we can see the promycelium and the septation and the formation of sterigmata, and the tip of the sterigmata swells and forms the basidiospores. So, basidiospores are formed after meiosis. So, it is haploid, and 2 of them are of plus strain and 2 of them are of minus strain. Here also we can see the formation of promycelium, septation, sterigmata, the tip of the sterigmata cell, and each promycelium produces 4 basidiospores. Here also we can see the germination of the teltospore. So, now we move on to the haplophase. This phase confined to alternate host Barbary. It consists of the haplomycelium and 2 spore stages, pycnidial and the ascidial stage. Basidiospore germinate on the leaf of the Barbary and put up a germ tube called primary hyphae. It infects young leaf of Barbary and form primary mycelium, also called monokaryotic or haplomycelium. Monokaryotic mycelium organized to form a dense mat under upper epidermis, and it forms the pycnidial stage. From the mycelial mat arises group of hyphae which organize themselves into pycnidium. So, here we can see this is the Barbary leaf. Upper portion we can see the pycnidial stage, upper epidermal layer we can see the pycnidial stage, and the lower epidermis we can see the ascidial stage. So, 2 stages are seen on the Barbary leaf. So, pycnidial stage, it is also known as spermagonial stage. It consists of flask-shaped structure, opens to outside by small pore called the ostiole, guarded by sterile hyphae. So, this is the flask-shaped pycnidial stage, and it consists of 2 structures, elongated receptive hyphae and also the spermatia producing spermatiophores. So, pycnidium or spermagonium, flask-shaped structure, opened outside by small pores called ostiole, guarded by sterile hyphae called periphysis. It may be of plus or minus strain depending upon the type of basidiospore germinated on the Barbary leaf. 2 kinds of hyphae in pycnidium: receptive hyphae, long, delicate, protrude beyond the ostiole; spermatial hyphae, short, arranged in a palisade-like layer lining the spermagonial cavity. So we can see the receptive hyphae and also the spermatiophore bearing the spermatia is seen a cell lining parasite-like layering on the spermogonial cavity. Spermatial hyphae bear spermatia or pycniospores at their tip. Insects make the transfer of spermatium from one pycnidium to another, that is plus pycnidium to the minus pycnidium plus pycnidial spermatiophore into the receptive hyphae of minus pycnidium of another. During the visit of insect, the transfer spermatium of one strain of pycnidial cup containing receptive hyphae of opposite strain. A binucleate cell is formed called a dikaryon. From this dikaryotic cell, dikaryotic mycelium is formed. So this is the pycnidiospermogonial stage. Here we can see the upper epidermis, the pycnidial stage, lower epidermis, ascidial stage. So next ascidial stage. So the fusion between the plus nuclei and the minus nuclei happens. From the dikaryotic cell, dikaryotic mycelium is formed, later organized to form ascidia in the mesophyll of barberry leaf. Ascidia are cup-shaped structure, inverted cup-like structure in which ascidiospores are produced. Ascidiospores are disseminated by wind and they gain access to wheat plant. Thus life cycle is completed. So each ascidiospores are almost around or some are hexagonal. At the young stage it is hexagonal in shape, yellowish red, uninuclear, binucleate, unicellular, binucleate cell, thick wall, 6 germ pores are seen, germinate on wheat plant. So it is an inverted cup-like structure. Here we can see the dikaryotic mycelium which produces chains of ascidiospores. This is a barberry leaf with a ascidial cup. This is the pycnidium and this is a ascidium. So coming to the life cycle, here we can see the ascidiospore, dikaryotic ascidiospores are coming and they germinate on wheat plant, produces the urinary stage. Rhodospores again causes a secondary infection. Rhodospore germinate causes a secondary infection and later in the same soray produces the teltospores. Teltospores with the dikaryotic nuclei fuses, fusion of the plus and minus nuclei, meiosis, formation of the promycelium and the formation of the basidion sterigmata. Basidiospores releases and they germinate on the barberry leaf and the haplophase started. Spermogonial stage, plus and minus. Spermogonial stage we can see the fusion of different strains of pycnidial and pycnidial receptive hyphae and the spermatiophore, and the resulting ascidial stage, inverted cup-like ascidial stage with the chains of ascidia. Again the dikaryotic ascidiospores germinate on the wheat plant. So we can see a dikaryotic phase completed on the wheat plant and a diplophase short-lived and a monokaryotic or haplophase which is completed on the barberry leaf. So this is the life cycle of Puccinia. Thank you.